We're going to take a look at how UV visible spectroscopy works. But before we do that, let's take a look at a general definition of spectroscopy. If the electromagnetic spectrum interacts with matter, then different things can happen. And if we understand what happens, we can understand information about the molecule. We're going to focus on visible, but we can look at what happens when we have infrared, radio, X-ray, or microwaves interacting with matter. So here's a picture of a simple spectrometer, and there's some sort of electromagnetic radiation which goes through a slit to narrow the beam, goes through a monochromator which helps us get to an individual wavelength, and it's basically like a prism. And then there's another slit, so only the wavelength we care about goes through the sample, the detector, and then is recorded. Now the visible spectrophotometer that we use doesn't really work that way. It's a little more complicated. It has white light going through the sample and to the detector, and then computer programs break everything down and separate the spectrum into the individual wavelengths. And you can see, as we've discussed before, that at one end of the spectrum, the X-rays and the gamma rays are very high energy, short wavelength. And at the other end of the spectrum, we have low energy, long wavelength radio waves, or extra low frequency waves. Right in the middle is where we're going to focus in the ultraviolet and the visible. We'll talk about these other types of spectroscopy a long time from now. If molecules absorb electromagnetic energy in the UV or visible range, electrons will change energy levels. We've seen that with the Bohr model of the atom. If we're in the ultraviolet range, then the electrons that are in the double bond, in a carbon-carbon double bond or carbon-oxygen double bond, will interact with the ultraviolet light. The other thing that can absorb ultraviolet light are proteins, around 260 nanometers. That's very useful if we're trying to determine whether or not a sample has some protein in it, a colorless sample. The visible spectrum focuses either on conjugated systems, and we'll talk about what that means in a few minutes, or the d orbitals of transition metals, ions. So we're going to focus mostly on the visible because our spectrophotometers only read the visible spectrum. So if we take a look at the color of light, what we see is the complement of what's absorbed. So if we see something that is red, it's absorbed the blue-green light. If we see something that's blue, that means the blue light is allowed through and the other light is absorbed, primarily the orange. And it's the light that's absorbed that is important in terms of looking at the energy and the wavelength. So you can see that violet is a shorter wavelength, higher energy, meaning there's a larger electron transition than if it's red light. So let's take a look at what conjugated means. In a conjugated system, we have alternate single and double bonds. So here is a small conjugated system. Double, single, double. And it absorbs in the ultraviolet. Its maximum wavelength is around 217 nanometers. Now let's take a look at one that has more conjugation. So here's our benzene ring, alternate, double, single, double, single, double, single, double. And we've talked before about how the benzene ring really isn't alternate, double, and single. It's really all those electrons spread evenly throughout. Well, it turns out that that happens through the entire molecule. And the longer the system of alternate or double and single bonds, or the system where those electrons can all be spread out and shared, the longer the wavelength. In order to really understand that, we need to look at a model of the atom that we're really not going to talk about. If you have more questions, I'll be happy to talk about it with you, but we're really not going to go into the details of why a shorter conjugated system has shorter wavelengths and a longer conjugated system absorbs longer wavelengths. But here's an example. So phenolphthalein in an acidic solution is colorless, but when we add base, it turns bright pink. So let's track the conjugation. In colorless phenolphthalein, we have one ring, we have a second ring, we have a third ring, and they're not connected by alternate single and double bonds. So we have single, single, single. Whereas when it turns pink, we have double, single, double, single, double, single, double, single. And we continue our way around, single, double, continues over here and over here. So this is a longer conjugated system than the individual benzene rings over here on the left. So on the left, light will be absorbed, but it will be in the ultraviolet region. We can't see it. In the right, light will be absorbed, and it looks pink to us. So let's take a look at what color is absorbed by the ultraviolet, by the phenolphthalein. So you can see here that phenolphthalein is absorbing in the sort of green-yellow range. So what we're seeing, so its lambda max is about 520, 40, maybe 550 nanometers. What we're seeing is the combination of the bluish purple and the reddish orange. And we see the bright pink, that's phenolphthalein. Now in the acid form, where it only absorbs in the ultraviolet region, it's colorless. It doesn't absorb anything colored, therefore we don't see any of the complementary colors. So here are a few important biomolecules that have conjugation. These are spelled a little strangely because they're British, but it's hemoglobin, which looks to us to be red, which means it's absorbing in the blue. We have hemocyanin, which looks to us to be blue, which means it's probably absorbing somewhere in the orange. We have chlorocrudrin, which is green, and so it's probably absorbing in the red or the blue area. And hemerythrin, which is violet, so it's probably absorbing in the red or yellow or green region. Remember, what color we see is not the color that's absorbed. And each of these has long alternating sets of single and double bonds, so there are conjugation. And also, interestingly, for each of these, there is a metal ion in the center. Iron, copper, iron, and iron. And the transition metal in the center is also important in terms of the different colors. So let's take a look at some inorganic ionic compounds and the colors that we see here. So we see potassium aluminum sulfate, alum, which in its powder form is white. A pure crystal of it is colorless. The reason it's white is because each of the individual tiny little colorless crystals has light bouncing in all directions. We have nickel sulfate, which you can see absorbs in the red and absorbs in the purple, no absorbance in the green, so it looks green to us. And we have copper 2 sulfate, which absorbs in the longer wavelengths, and we see the shorter wavelengths. So for each of those transition metals, we see a colored compound. For potassium and aluminum, which are main group, so in columns 1 and 13, those main group elements, those are colorless. And this is why. If transition metal ions are surrounded by water or other small molecules, the d orbitals split. So that's probably new to most of you. The d orbitals in an atom are typically all at the same energy level. Maybe they're 3d electrons. But if they end up surrounded by water, then what happens is the individual orbitals will split. And whether they split into a 2-3 pattern or a 3-2 pattern, or sometimes a 1-2-1-1, there's lots of different patterns. You will not have to know them. Whether or not they split and how they split, that depends on what is surrounding the central cation. And the distance is also determined by what's surrounding the central cation. And the distance between the lower and upper d orbitals tells us the wavelength. Longer split, more energy, higher wavelength. Closer together in energy, lower energy, longer wavelengths. But if an electron moves up here, that happens when light of the correct wavelength is absorbed. If light's absorbed, then we see all the colors that are not absorbed. The main group cations, so those are the group 1, group 2, group 13, are aluminums and boron and potassium and sodium. They do not absorb visible light. And we can see here, this is a picture of a metal ion surrounded by water molecules. And because the transition metals have partially filled d orbitals, some become higher in energy, that's what I just showed you, and electrons move between the higher and lower d orbitals by absorbing a photon of light. The absorbed light helps us with what colors we see. And so these different charges, or oxidation numbers actually, produce different colors. So vanadium could look to us to be purple, green, blue, or yellow. Iron might be green, it might be yellow, or sort of a yellowish orange. Chromium, we can have blue, green, orange, or sometimes even a yellower color. So these transition metal ions are all different colors because of the change in d orbitals. So we've seen two different things happen. We've seen energy absorbed because of long conjugated molecules. Those tend to be our organics. And we've seen energy absorbed because of d electrons. Those tend to be our transition metals. In any case, both of those can be used in visible spectroscopy to try to understand more about an atom or an ion or a solution. And we'll talk about that more in the next video.